you and welcome. So, thank you for coming here to listen to me and to Diamyd Medical. We are a company in the diabetes space and especially focused on autoimmune diabetes, where type 1 diabetes is the most known indication in that space. We're a company with a purpose to cure the disease. That's always been the purpose of the company ever since it was founded. And it was founded when the founder's youngest daughter got type 1 diabetes. So the purpose has been always to cure the disease. And I will today present the company, but with a focus on the very latest results that has been really, one could say, a breakthrough for the company. This is obviously important, but it's boring, so let's go to the next part. This is the cliffhanger, and I will come back to this, but just to keep your interest up, this is what's really been a breakthrough for us as a company, and it has de-risked the development in the company. In December, we press released two major things. One, extensive meta-analysis from previous studies, including a European phase 3, a US phase 2 trial, and a Swedish phase 2 trial, where we had a hypothesis that certain genotypes may affect the response to the diabetes vaccine, which is our lead asset, and indeed it does. So we've really shown that we identified the patient responders already. And the second one is also in December, where we have evaluated a new administration route in a pilot trial, and based on the results, it really looks like that new route significantly enhances the effect. So two really major events that has de-risked the development for our company. And I will come back to this. Type 1 diabetes. So you have type 1 diabetes, you have type 2 diabetes. If you simplify, type 1 diabetes is an autoimmune disease. So the immune system destroys the insulin producing cells in the body, and you become dependent on insulin for the rest of your life. It's a prevalent disease. There are more than 40 million people who have this disease in the world. Most importantly, there are no disease-modifying therapies available. There's only insulin, basically. And we've had that for 100 years. We've become very good at insulin, and it's saved millions and millions of lives. But it's time to get something to really start addressing the underlying disease here. The incidence, it's most common in children and young adults. It's increasing every year. What's maybe not that known is that, there's a significant unmet medical need here. Based on a very recent Swedish register study, it shows that if you get the disease diagnosis before the age of 10, you have a 14 to 18 year shorter life expectancy compared to non-diabetics. And obviously there are significant healthcare costs involved in diabetes. And interestingly enough, which is also not very well known, up to 10% of type 2 diabetics have autoimmune diabetes. They are today diagnosed as type 2 diabetics, they are treated as type 2 diabetics, but they are actually more like type 1 diabetics. So they should be treated like type 1 diabetics as well. And this is for us really interesting because this opens up a big portion of the type 2 diabetes market for us as well to go in with a disease-modifying therapy. This is interesting, and this will tie back to our subgroup analysis that I highlighted in the beginning. This came in January. It's a discussion paper. A lot of the key opinion leaders in the type 1 diabetes field is here as co-authors, where they said that it's time to start thinking precision medicine in type 1 diabetes. We can't expect that one single drug will sort of work in everyone, which is common for all complex diseases. You never have anything that works in everyone. And they identified that there are probably two subgroups of patients, main subgroups, that are defined based on their autoimmunity. You have the GAD autoimmunity group, which is a HLA DR3-DQ2 genotype that drives this autoimmune subgroup. And then you have the pro-insulin autoimmunity group, which is a different HLA genotype that defines that group. I can say that the subgroup where we show that our asset, which is based on the GAD molecule, works in the GAD autoimmunity group, which is around 50% of all the patients that we've had in our trials. So these are our two assets, the Diamyd, which is it's a recombinant protein formulated in alum, It's one of the antigens that the immune system recognizes as foreign and goes to attacks. It's a first-in-class therapeutic. It's based on, and it's in phase 2b, that's our flagship, and it's been in phase 3 before, so we have a lot of safety on this asset. It's been in more than 1,000 patient trials with more than 1,000 patients, never any serious adverse events. Second one is a small molecule based on GABA, which is a cell signaling molecule. Preclinical work shows that it can regenerate insulin-producing cells. We have our own formulation of this now, and it's in clinical studies as well. We are focusing on intervention in type 1 diabetes, which means newly diagnosed type 1 diabetics. We are now going into autoimmune type 2 diabetes as well with a clinical trial. And prevention is a big part as well of type 1 diabetes today, where you can identify individuals who are at high risk of getting the disease, and that you can prevent them from, or at least prolong the time to diagnosis, or in the best case, prevent them from ever becoming diabetic. And obviously regeneration is something that you would need in the end to fully cure the disease, to regenerate the insulin producing capacity, and that's the aim of our second asset. And this is also important, and then how do we position ourselves? So we really want to develop something that is both very convenient and safe and has the best possible effect. So we've shown with the Diamyd diabetes vaccine that we have very good safety, and based on the results we have today, we show also very good clinical effects. We don't want to develop something that is either a very expensive and very exclusive treatment that people who only have access to very exclusive hospitals can. We want to make sure that we can reach as many patients worldwide as possible. This is a simplified version of the mechanism. So GAD, which is the active component, the protein. And this is what happens in the body naturally. Like antigens, proteins are taken up by antigen-presenting cells. They are presented to T cells. Part of this complex that presents antigens is the HLA complex, so major histocompatibility complex, so which is interesting given that we've now shown that depends on which genotype you have that codes for these HLA molecules does affect the efficacy of our vaccine. And what's discussed generally in the field is if you want to create immunological tolerance, as we want to, you can either go through create T cell deactivation, you deactivate the cytotoxic T cells, you create T cell exhaustion, or that they actually die out, or you induce the regulatory T cells that sort of keep a brake on the immune system so that it doesn't create autoimmunity, or you modulate the effect of the existing T cells so that they become more anti-inflammatory and tolerogenic. And we actually see based from our clinical trials now that we do see patterns that look like the T cell deactivation, exhaustion, and also T cell modulation. And we get this, if you're more into immunology, we see in the IL-10 cytokine signal coming up around 1 year after baseline, which is an indication of that we've induced tolerance against the active component. So first, when it comes to the subgroup analysis, so this was more than 500 patients included in the analysis from previous clinical trials. And the hypothesis was that we believe that HLA influences the effect of the vaccine, and indeed it does. So we see a clinical effect ratio of up to 60%, highly significant compared to placebo in this group that is also called GAD3, which is then defined by autoimmunity against GAD, which is the active component in our vaccine. And this really supports a precision medicine approach in autoimmune diabetes, and it does de-risk the development for us because we've sort of shown that our previous clinical trials, even if there were some question marks then and didn't really meet the primary endpoint, we now show that, around 50% of those patients actually get a really significant effect of our vaccine, and there's a dose response as well. More injections give a better effect. And then secondly, this new administration route, which comes from the allergy field, where they tested that rather than injecting subcutaneously, you can inject directly into the superficial lymph nodes and you get a significantly better effect, or you get a much faster effect In allergy, they usually use immunotherapy for 2 to 3 years to desensitize the body against an allergen. But then they've shown in clinical trials that it's enough with 3 injections in a lymph node, you get the same clinical effect. So we tried the same thing in an autoimmune disease, and based on the pilot clinical trial where we now have the full results, it really does look like that it enhances the effect quite considerably. And these are the results, and I will try to pedagogically explain this. Top left-hand corner, that's the most important efficacy parameter, regulatory. So it's a measure of the endogenous insulin production. How much insulin does the patient produce? The red line, that's a comparator group. This is an open label trial, so we've used this from a previous clinical trial patients that have been placebo treated in the same age group to show what the natural disease course looks like. So usually the patient reduces about 40 to 50% in the insulin production over the time of 15 months. Our patients at 15 months have gone down around 18%. At 30 months, we don't have any comparator groups with that long follow-up anymore. Our patients have gone down around 40% on average. Interestingly, 3 patients have received a 4th booster injection, and they actually increased their insulin production. And 1 year after the booster injection, they are still at the same insulin producing capacity they were a year before that. So we've been able, with a booster injection, to prolong or preserve the insulin production by 1 year. So this really opens up the possibility to have booster injections in the future, maybe annual boosters, to prolong the preservation of the insulin production. Below that, you see the exogenous, how much insulin do the patients need to inject to maintain their blood sugar values. Usually you have to increase your blood insulin injection, like the dose, quite considerably over the span of 1 to 2 years. Our patients have not, on average, increased their insulin production after 15 months, and after 30 months they are much below what could be expected. And again, interestingly, the 3 patients who got the booster have actually reduced their insulin production, requirements further. And we see that effect is even at around 42 months, they are still below baseline. And then the blood glucose values, or HbA1c, that's the most important clinical parameter that the physicians treat to target the patients need to keep below certain range. Usually it's very difficult to do this, so patients keep increasing in their HbA1c. Our patients go down, and then they actually stay below baseline for 50 months, for 30 months, and the 3 patients who got their booster injection further go down in blood glucose. And the partial remission, which is in the upper right corner, that's a combination of the exogenous insulin requirements and the blood glucose. So when you combine those two in a simple equation, you call it partial remission if you're below 9. And all 12 of our patients are in partial remission at 15 months, 9 out of 12 at 30 months, and the 3 patients who got the booster injection are all in partial remission still at 42 months. So this is really promising results, and we have a phase 2b ongoing, which will read out the 3rd quarter this year, 109 patient, placebo-controlled. This is our pipeline then. So the results I showed was from Diamyd 1, the 12 patients. Diamyd 2, like I said, that will read out 3rd quarter this year. We are starting together with the Norwegian University a trial now in autoimmune type 2 diabetes, and LADA, which is also called. And then our second asset is also in clinical stage in a phase 2A, where we have the first dose escalation is finished. Safety looks good. We are waiting for the first metabolic results, and the second stage has already started in that trial as well. Financials, we have cash enough to go beyond the results from the phase 2b trial, so all the ongoing activities in the company are fully financed. And we have a market cap which has increased quite significantly now, obviously with the results we've shown recently, and it's around $130 million US. Board and management. I want to highlight especially the founder, Anders Möller, who's a director of the board, and he's still very active in the company. And also Mark Atkinson, who joined our board a bit over a year ago, who is one of the really biggest key opinion leaders in type 1 diabetes. We're very proud to have him on our board as well. And obviously I'm very proud of all my employees here. Fantastic team. And to summarize, significantly de-risked development now in the company because of the subgroup analysis and the clinical results we show. We have 2 unpartnered first-in-class clinical assets for type 1 and autoimmune type 2 diabetes. And there's a very high unmet medical need in this indication. It's obviously a blockbuster market as well. And if you have any questions, you can ask them now or contact me by email or just grab me after this session. I'll be glad to answer any questions. Thank you.